to last in nursery and the trees. So if you look into the losses caused by this pathogen, so the losses due to the diseases range from a few to 80%. And in case of a severe infection, there is 100% crop failure. And you know that how this disease develops. For development of the diseases, so there should be a susceptible host and a virulent pathogen and a favorable environment condition. All the three, if it is existing, then the disease will occur. So the management part, we should always consider what type of a host we are using, whether it is a resistant host for cultivation or plantation, or what type of pathogen which is existing, whether it is a weak pathogen or virulent pathogen. So how to control these pathogens, and favorable environmental condition by altering the time of sowing and planting, thereby the plants will escape from the pathogenic attack. So with this introduction, we can see that the days after planting and the population of disease plants, disease over a time. If you look into this crop and the days after planting, if it is increases, the susceptibility of the host will also increase, and thereby the disease, it is highly prone to the disease. And look at these trees, and you can see that juice here on the right side and the juice here on the trees, and here there is an infection, and you can see there's another polyporous fungus which is attacking the wood logs. Also, in another slide. Yeah. This is a bacterial infection on the trees, and you can see this, it is known as fire blight. And you can see this, all the, these leaves which is showing there. And here this is Sisophyllum infection on the wood, and you can see this split gill fungi, which is known as Sisophyllum commune, which is vastly attacking the tree species. And this is, of course, the bacteria which attacks the trees and gives the appearance of fire blight. And if you see this, the nursery, and after sowing it, most of the plants are susceptible to this damping off, and you can see this after sowing. And there is, if you sow in a tray or if it is in the field, and it causes this infection. So these nursery disease already I have told you, and that you can watch another video that I have explained about these diseases. And these diseases, it is more prominent in the nursery level. And you can see here, this is a neem nursery, which is attacked by other fungi. There is a Potemkin fungi, which is attacked on the leaf part. And also further, you can see here these diagnosis. So before management, we must have an acute diagnosis, whether it is a fungal or bacterial or virus, and then monitoring the pathogens. And it is very important. If it is for fungi, it produces a specific symptoms on the leaf, stems, branches, and sometimes rods, and thereby we can identify it is due to this fungi. And moreover, for the bacterial pathogen, it is an easy test we have, that is a Woos test. So if you take out the infected plants or the stem and cut it and dip it in a clean glass of water. So when you do this, dip it in the clean glass of water, the bacterial oozes will be coming out. So thereby we can identify that it is due to bacteria. And if it is in the case of virus, it produces a lot of mosaic symptom, and then curling, puckering, all kind of symptoms. Based on this, we can identify it. Okay, so this acute diagnosis of a disease is very important. And then after identifying, So we have to monitoring the pathogen, which is how extent it is that. Okay, so based on that, we can initiate the management method. So the concept of integrated disease management, where the disease are managed by integrating range of control methods and practices. So we are going to see different kind of methods like cultural, mechanical, physical, all methods integrating all the practices, and then we can manage these diseases in the nursery as well as in the forest. IDM calls for a minimal use of pesticides or fungicide, and only if deemed necessary. If it is necessary, we can use it, giving the preference to control methods such as host plant resistant, like selection of resistant varieties, and other cultural practices and biological methods. An IDM, we can define it currently as a sustainable approach to the managing disease by combining biological, cultural, physical, and chemical tools in a way that minimize economic, health, and environmental risk. With this, we can, that is the definition. And I'm going to talk about the integrated disease management in the following component. Like the first one is cultural methods, and then mechanical methods, and physical methods, chemical methods, and biological methods. So briefly, I will explain about this. The first one is cultural methods. Okay, and then subsequently we can see this mechanical method, physical methods, and the chemical methods, and the biological methods. Like this cultural methods, I can say this is a no cost. We are not going to put any cost in it. Okay, so that is a very simple method by which we can manage the disease. Here, this is a selection. So, for example, selection of resistant varieties, selection of site, and it is playing a major role. And then field management, okay, like field sanitization, removal of fallen leaves or the debris from the field. So, and then cleaning, and then removal of infected trees or dead trees. Okay, so the removal of fruiting bodies, all it comes, and then eradication. And then here we must eradicate. It means we have to remove that fruiting bodies, and we have to eradicate them. And if you see here, that is a decision making. It is also very important. Like what is the best time to show this crop? Okay, and then what is the best time to manage this disease? All we can do it. Okay, so that is the decision making. Here you can see this on the wooden woods, and we can see this latiporous sulfur fungi. Okay, so we must eradicate them. The first one is a disease management by cultural method, and in this method, so first we have to remove that fruiting body which is grown on the wood. Okay, so we must carefully remove these fruiting bodies, and we must put it in the polythene bag inside the polythene bag and tie it properly, and then we must throw it, we must burn it, bring out of other place, and then we have to burn it. That is a very important. Okay, and then we can see here this is removal of collapsed trees. So if you have any trees which are totally collapsed, we must remove them. That is that will harbor the pathogen, and better to remove this collapsed trees from the forest area. And you can see here also, and we must remove this here, this neem trees which infected with this mycoplasma, and there we have to remove these infected trees, and thereby we can save other trees. And here also you can see this infected, if any basidiocharp or the fruiting body or fungi, so we must remove them. Okay. If you see this base of the tree, there is a Ganoderma infection, and we must remove these Ganoderma fruiting body, put them into this polythene bags and throw it. That is very important. Okay, and then we can see here, this is a removal of Loranthus. Any parasite, it is there, any epiphytes or parasites on the growing tree plants that weaken the host. So we must remove these host, that is parasitic plants from the trees. So here parasitic Loranthus, it is easy to identify. You can see this one basic stem. From that multiple shoots maybe arises. That is easy to identify this Loranthus species. And then, you have to remove this Loranthus and then we must throw them or we must burn them. Okay. So, so next one is application of organic manures. So organic manures, we can, collect this organic manures like farmyard manures and then vermic compost that if you apply in the nursery and then even in the forestries that grows very nicely. Okay. And then soil amendment. And then crop growing season, okay, which is the best season that we can choose, that we have to identify that particular season, then we have to sow. Okay. Then so that is sowing date is very, very important. It differs from one tree species to another species. Okay. And then adjustment of sowing time, it is very important. Right? And then that if it is the first week of October, we must sow the plants in the first week of October. Okay, and then growing of seed crops and selection of the seeds and the seed material is very important. Whether this seed material is free from any infection, that we have to observe it, any bulbs or any tubers, any culms. Okay, so we must observe them and then we have to eliminate them initially. Okay, and then leveling the field and provision of drainage facilities, it's very important. The nursery level, we must you can see this background pictures, we must prepare the leveled beds for raising the nursery. Okay, so that is very important, right? If you grow in a low-lying area like waterlogging area, here you can see in this picture, and if it is a waterlogging area, it will kill all the plant species. So we have to choose the site is very important, and that should be of free from waterlogging area. Otherwise, that will favor the pathogenic attack. And then seed rate. So if it is in 1 unit area, if it is 100 gram, 1 meter square, so we must sow only 100 gram per meter square. If you sow more seeds, and then it will create the problem. And burning of stubbles and crop residues, collect all the stubbles and then burn it. And then depth of the sowing is also very important. It is depends upon the size. If it's a seeds is very small, that we have to, within 1 centimeter, we have to show 1 or 2 centimeter. If the seed size is very big, then we have to sow in a deep area, right? And then, that will, protect the seeds. And then spacing is very important. Okay, so when you do the sowing, spacing is very important. If you do the closer spacing, then there will be a competition for the nutrients and the host will become very weak and it will be prone to the disease. So these are the points we must remember. And then close spacing. Say, for example, we here, this is a citrus tree, and you can see this. If there is a close spacing, it will not give any fruit at all because of competition for the nutrients, and then lack of minerals in the soil, or in lack of minerals because in a unit area there are 4 or 5 trees, so thereby the competition will be there for the nutrients, and it will not produce sufficient hormones for fruiting and flowering. Okay, so that's why closer spacing, it is affect the trees. Okay. And then do the approach grafting. If it is infected, and select grow this another supporting seedling, and then do this approach grafting. Thereby it will help the trees to grow. Okay, and then you can see this. It is more useful for half dead trees. And then methods of sowing planting. It is very important in which method you are sowing. And altering the soil pH is also very important. And then mulching, so covering up to preserve the moisture. And irrigation water management is also very important. If you give excess water to the nursery beds, and that will create a problem, then damping of disease will be occurring on it. So then field and plant sanitation also, it's very important. And roguing out. Okay, roguing out the infected plants, it is very important. Okay, so field sanitation, like sanitation. It's a removal of all the dead branches or fallen leaves, so that we must properly remove them. And if it is any weeds, that we must remove them. It is very important. Okay. And then mechanical method. This mechanical method, it is selection of after selecting the seeds. So we must remove this infected planting material seeds by electronic color charter, or mechanically you can remove them. Okay, you can sort it out, and then using 10% sodium chloride, you can use this method. Is using a 10% sodium chloride. If you put in this bucket or beaker, you can take, and you prepare the 10% sodium chloride. Take 1 liter of water, and you can add 100 gram of salt. So it gives a 10%. So if you take a 10 liter, then 1 kg. Okay. So then after that, you have to mix it well. The healthy seeds will settle down, and the infected seeds will be float on the top. Thereby, we can remove all the infected seeds. And the next one is a physical method. In this physical method, that is, heat is involved, hot air or hot water. Okay, hot air and hot water basically used for treating the seeds and the soil. Okay, no, it is not used in the trees or in the forest, because a hot air, we can sterilize the soil. Because the soil-borne pathogen used to be reside in the soil. So this hot air, if you pump this hot air, and all the pathogen will die. And if you pour the hot water, pathogen will die. And hot water, it is useful for treating the planting material. Okay, basically to kill the virus. And then burning. Okay, second one is burning, and the soil solarisation. Also one by one. And in the physical method, hot water treatment, hot air, and aerated steam therapy. There are three things. Hot water directly you can pour on the nursery bed, thereby the pathogen will be dying. And then hot air, you can pump this hot air using the blower, generate the hot air and pump into this soil. Or you can keep some planting material, pump this hot air, set this 50 degree, and then pump into the closed container. Keep the planting material in the closed container and pump this hot air. Thereby, you can eliminate all the pathogens. And aerated steam therapy also, the same thing, in a closed container. Keep all the planting material, pump this aerated steam therapy. Make sure that you are always maintaining 50 degree centigrade. Then you have to, for every planting material, there is a specific time limit is there. So with this, we have to manage the disease. Okay, so the first one is method, and in this method you can see this is spread all the leaves in the nursery bed and then burn them. Okay, and if you burn, what will happen? So it will catch the fire, then the soil temperature rises even 60 degree. Okay, then all the pathogen which are there in the soil, it will die. Okay, you can, if you have a thermometer, you can check them, and that all the pathogen, it will die. If you have thermometer, you can check, it is around 60 degrees centigrade. Okay, so thereby all the pathogen which are there in the soil, it will die. And then after that, we can go for sowing. Okay, so the next one is a hot water treatment. Here, tubers, bulbs, sets, and any kind of planting material, dip it in the 50 degree centigrade for 20 minutes. Okay, so after boiling, keep the container outside, and then sure that it should be 50 degree. It should not be more than 50 degrees centigrade, and you have to keep it for only 20 minutes. Thereby, we can kill all the virus, which are bacteria or virus which are there in the planting material. We can kill them. Okay, and then solar heating. Okay, using the natural heat, we can kill all the pathogens. Okay, so we can trap this solar heat. Okay, so when you cover this polythene sheet, okay, I'll tell you, the temperature may increase up to 5 degrees centigrade. Okay, so the 5 degrees sudden increase in temperature, all the microbes which are there in the soil, it will die. Okay, so thereby eliminating all these pathogen in the soil. But sometimes this a high temperature favors antagonistic fungi. There is a beneficial fungi which live in the soil. Okay, so due to sudden increase in temperature, all the pathogens like Fusarium and Pythium and Verticillium, all it will die. Okay, so that's what is happening in this method. Solar. In this picture, you can see here. So prepare the nursery bed and cover it with the polythene, and then you can cover it with a polythene and you can keep it. Okay, so thereby, so we have to keep it for 7 days. Remember, we must keep it for 7 days. So all these, this pathogen which are there in the soil, it will die. Okay, so the fourth method is chemical methods. In these methods, separate for each diseases, specific chemicals are there that you can learn in other chapter. And now this is a seed protectant. Okay, if you want to do any seed treatment, so we can use it Capthon, Therum, Carbendazim, and Carboxyl. And the recommended dose is 2 gram per kg of seed. If you have a 1 kg of planting material, you can use 2 gram. That is a seed protector. Soil fungicide. If you want to kill the soil-borne pathogen, we can use this borax mixture or copper-oxychloride or chloropicrin or formaldehyde or vapor. These are this. For borax mixture, 1%, copper-oxychloride, 0.2%. So chloropicrin is chloropicrin, formaldehyde, vapor, or soil fumigants. Okay, so that I will explain it later. Okay, so this you must understand. So these borax mixture and copper-oxychloride, it can kill the most of the soil-borne pathogen like Fusarium, Verticillium. Okay, and then soil fungicide, borax mixture, and copper-oxychloride. Okay, and then foliage and blossom. If the disease occurs on the foliage and blossom, that we can use a Capthon, Ferbum, Zinap, Mangosap, Chlorothalonil. Okay, so Capthon, Ferbum, Zinap, it is the recommended doses 0.1%. Okay, and then some kind of a fruit protector. If a fruiting stage is there, though we can want to protect the fruits from post-harvest pathogen, so we can use a Capthon, Manap, Carbendazim, and Mangosap. The recommended dose is 0.1%, 1 gram in 1 liter of water. And then eradicants, lime sulfur. Okay, so if you spray lime sulfur, and it will eradicate all the pathogens which are there in the surface of the leaf. Okay, and then tree wound dressers. If the tree is wounded or any kind of polyporous Ganoderma attack is there, so you have to cut and remove all those fruiting bodies are there, and then we have to apply this borax paste or Chopathia paste. Okay, so these are the different kind of fungicides which are available. Okay, and then take example of antibiotics, streptomycin and tetracycline. It's basically use streptomycin to kill that bacterial pathogen, and a tetracycline like mycoplasma like organism. I have shown in the previous slides that is neem yellowing that is caused by mycoplasma like organism. There we can use a tetracycline 500 ppm or streptomycin. Commonly, if you find any bacterial disease, that we can use streptomycin 500 ppm. Okay, and then antifungal antibiotic, aureofungin. This aureofungin antifungal antibiotic. Aureofungin, it can kill this Ganoderma. Okay, so this kind of a lot of antifungal antibiotics are there. Okay, and then how we can apply these chemicals into the soil, or how we can apply on the foliage and the blossom. There are different methods are there: foliage, foliar application, soil drenching, broadcasting, furrow application, soil fumigation, and chemigation. So what is a foliar application? So fill these required chemicals at, as I said, Capthon, Carbendazim 0.1%, and just you can spray it. Okay, so Carbendazim 0.1%, you can, or copper-oxychloride 0.1%, you fill it on the tank and then you spray it on the trees. So thereby we can protect the trees from the pathogens. Okay, and so the foliar pathogen, say for example here Helminthosporium tursicum. If you want to control this Helminthosporium tursicum, so we can spray this copper-oxychloride 0.5%, and if you spray all these pathogens, it will die. Okay, and then sooty mold disease. If you see bamboo, bamboo is largely infected with this sooty mold. It is fungi which covers epiphytically on the leaf. And if you spray that maida 5%, and this is a sticky substance, it will eliminate all the, it is sticking to the leaf and it will dries up after some time and it comes out as a flakes. So management is a foliar spray with meta 5%. Okay, so thereby we can manage this sooty mold disease. And then soil drenching. So required amount of fungicides after preparation, we just drench the soil. Okay, if a sealing is there or any kind of season, just you can drench it. Thereby we can manage the soil-borne pathogens. Okay, and this is also an efficient method. And soil fumigation, as I said, soil fumigants in one slide. This formaldehyde, vapor. Okay, so these are all these soil fumigants. So just spray or apply it on the soil and then spread it with polythene bag immediately. Okay, immediately. So thereby these fumigants, it will percolate into the soil and it will kill all the pathogens which are there in the soil. Okay, so thereby we can manage this. Cover it for at least for 3 to 4 days, and then after that you remove this polythene bag and all the fumigants, it will escape. Then only we have to go for sowing. Okay, so that is a soil fumigation. So this way we have to cover it, or we can temporarily seal it with this sand or soil, keep the stones on the corners, and thereby we can this formaldehyde will be retained on the soil and it will be managed efficiently. And this borax paste 10%, we can cut this cut into wounds, or if any kind of infected branches are there, remove them, or any gummosis on the trees, scoop it out and apply this metalaxyl or this borax paste 10%. Thereby we can manage this gummosis. Okay, and then foot rot and dieback symptom after pruning. You can apply with this metalaxyl, or we can use this borax paste. Okay, so with this we can manage the disease. So this borax paste also we can apply on the trees systematically. You can apply it on the base of the trees up to 1.5 meter level, and there it will protect all the pathogens. Okay, and this is the you in this way you can manage. Okay, and then dry seed treatment. So before sowing the seeds on the nursery, we can use this to carbendazim 2 gram per kg and mix it with per kg of seeds in a plastic container. After mixing it with for mixing, we have to add a little bit of water. So thereby these chemicals, it will be coated uniformly on the seeds, and we have to dry it for 24 hours under the shade, and the next day you can take up the sowing. Okay, so these are the chemical methods. Now coming to this last one, biological methods. The methodology which I said earlier, all the chemical methods we can follow the same thing, but they replaced with this biocontrol agents like Trichoderma viridi. It is a pest fungi, so it can kill most of the soil-borne pathogens, Fusarium oxysporum, and then Verticillium, Pythium fatatar, and so on. Okay, so seed treatment, the recommended dose is 5 gram per kg, and the soil application is 5%. Okay, so seed treatment 0.5%, soil application 5%. Okay, so the same methodology which I said earlier, seed treatment, 5 gram of Trichoderma or Pseudomonas putida, mix it with 1 kg of seeds, and then add a little bit of water and then stir it well, and then you have to dry it under the shade, and then you can take up the sowing in the next day. Whereas in the soil application, mix a 2.5 kg of Trichoderma formulation in 50 kg of farmyard manure, mix it well, and then apply it in the soil. Okay, it can kill most of the pathogen which are there in the soil. Okay, so with this we can manage the soil-borne pathogens. Biocontrol method is the best method, and it is eco-friendly method that is recommended to manage all the diseases. Okay, and thereby we can biocontrol methods, if you employ, you can manage the soil-borne pathogens, and the seedlings will be more healthy. So here you can see in this barrow, there's a lot of seedlings are died due to this pathogens. Okay, so if you apply this biocontrol agent 7 days prior to sowing, and it can multiply fastly and occupies the space, thereby it will protect the pathogens. It will protect the host from the pathogens. Okay, so these are the pictures I just want to share with you. We have visited the field. Thank you for watching. Thank you.